people to serve in the army and the military. Here is one such person. Yeah. So can you just imagine this guy was just 21-year-old when he got into service. He went to Vietnam. He served there in Navy, U.S. Navy, as a hospital corpsman. He was there for a year. And during that period, when he saw so many people, wounded and they were about to die, he even, thought of committing suicide. He went into the seas, swam for some period, saw a shark, then he came back, and then he returned to the base. Then, after he came back to US, and he went for studies, and he decided to do medical sciences because he could realize that if he has better knowledge in medical sciences, he can do a lot of good service to people who are having problems. Okay, so this is the guy, and he said before he went to Vietnam, he was only getting C and D's in schools. He was a very average, below average student. But then his learning changed because two of his professors, English professor and a chemistry professor, they actually encouraged him to learn how to learn. So that was a turning point in his life. Okay, it's echoing. Okay. So, can you recognize this guy? Any one of you have seen this picture anytime? No. Well, so that means we are all beginners to molecular biological research. So many of you, some of you would have listened to his,
Yes, an institute by his name.
Can you identify this man by name? Well, of course, this picture you could not identify, no, I'm very sure. It's very impossible. But at least this, no way. Right. So we are now talking about Greg Wender, okay? And this is the guy who is well known for many contributions. And one of his contributions is about genome sequencing of bacteria. Back in 1995, he was responsible for 2 publications in Science, and that gave us rapid sequencing of bacterial genomes. And he also contributed several others, which I will show you during my talk. But this contribution is something very important. Also, in his name, there is an institute called JCVI, J. Greg Wender Institute. So initially, he started as the Genome Research Institute, and then it changed. Later on, he also established a JCVI. And when he published 2 papers, that was from the Institute for Genome Research, TIGR, and he could sequence 2 complete sequences of bacterial genomes of Haemophilus influenza and Mycoplasma genitalium. And these 2, and one is about 1.78 megabase per, the other one is in 0.57 megabase per genome. And these were the 2 important publications in that time because this was the first report about genome sequencing of bacteria. Right, and then there are attempts to compare these 2 genomes, and also they constructed minimal genetic complement of genes, number of genes which are required for any bacterial organism to be alive, minimal set of genes which are required. And they identified as 250 genes, but that was not the work done by Greg Wender. And because of his reports, people started having several other organisms sequenced, including archaean, Methanococcus, currently known as Methanococcus jenense, and this is a methanogenic archaean, and also Cyanobacteria cyanococcus. So from within 4 or 5 years, everybody started sequencing bacteria. So he was the man who was responsible for bacterial genome sequencing. Right. These are the 2 important papers. I'm just showing you, and I'm rather asking you to read this paper because they are really important even now. The papers which have been published in 1995. Those 2 appeared in Science in volume 269 and 270. Okay, and this one is about the whole genome random sequencing of Haemophilus influenza. The other one is about minimal gene complement of Mycoplasma genitalium. These are two important because before then, we had a genome sequencing of bacteria phage reported by Frederick Sanger. You could see this man, old man, and of course he got his first Nobel Prize when he was 40 years old, and he got a second Nobel Prize when he was 62. That was for DNA sequencing. So the first one is for protein sequencing. So he is one of the two scientists who have been awarded 2 Nobel Prizes in chemistry. He is the only one. There is another guy. He is, I think, Barry Staple. I forgot his name. Second man is also for the chemistry. But before then, we have Madame Curie, who got a Nobel Prize in physics and chemistry, 2 Nobel Prizes. Okay, what he did is he introduced, chain terminating sequencing method. So Sanger's DNA sequencing method. So all of us became very, curious about it, and also we started using this methodology. So everybody started using it. But how to prepare those genome for sequencing? That is a trick. There, in that time, they introduced 2 methods. One is hierarchical short sequencing, also called clone by clone method. The other one is whole genome shotgun sequencing. That was the one which was introduced by or applied by Craig Venter. So that is why bacterial genome sequencing became so easy. So if you read any textbook or maybe some of the simplest version of these 2 methods, you will see pictures like this. Hierarchical shotgun approach, where the mapped DNA will be fragmented into clones, and then clones are fragmented, sequenced, ordered, and assembly. And whereas in whole genome shotgun approach, the genome of the organism is fragmented into pieces, and then they are sequenced, and assembly is done by computer algorithms or softwares, right? So that is not so simple when it comes to working. So we have, there are basically 4 steps which are involved in clone by clone method, or hierarchical shotgun approach, where we have to prepare the back clone. What is back clone? Bacterial artificial chromosome. So clone is used for taking up the fragment of the DNA which is available, which is prepared from the genome of an organism. Then prepare a clone fingerprint. Then do the back clone sequencing, and then sequence assembly. That was a little cumbersome because it is a time-taking process, and it was too much lengthy also. So till that time, 1977, when Dr. Sanger reported about the sequencing of phage 174 bacteriophage, till 1995 there were no reports about genome sequencing of any microorganism. Almost for 20 years there are no reports. So only by Craig Venter the sequencing, we could get some knowledge about the bacterial genome, right? So that was actually by shotgun method, where they will isolate the DNA, random fragmentation of the genomic DNA, then do the site selection by electrophoresis, then library construction. That was again parent sequencing, then genome assembly was done. This is one, I'm putting it in a very simple term, but some of you would have at least attempted the nanopore sequencing in the laboratory, or if if you have an access to Sanger machine, you can also do this sequencing. But all these things are being done by automated version. Manually, it is not that easy to do sequencing. So we have now sequencer which are done automatically. So that is an advantage. So put the whole thing in a simple manner, we have a bacterial culture which is DNA is extracted, and then it is sheared, then the library is prepared, the library is used for sequencing, and then the whole sequence is assembled. So that is the flow chart which is available to us to prepare the sequence, right? So back to this 1995 paper of Craig Venter's group. The whole genome random sequencing, they also given this sequencing strategy. So it is available right in the paper. They also mentioned about some of the important features of the whole genome sequencing. So they said how many, number of double standard templates they could get, then what they even, what are the roles of those DNA information which they can assign for amino acid metabolism, cell envelope, cellular process, fatty acid, energy metabolism, and then regulatory function, all those things, they were able to do it in 1995. And this is the gene map of those organisms, which is also given in the same paper. And then there is also a circular representation of the genome, which are actually the genes have been predicted, where exactly those are there. It's all given in this paper, right? And the other paper is a minimal gene complement of Mycoplasma genitalium. There also they gave the gene map of the organism, Mycoplasma. And then they did the comparison between the Haemophilus influenza and Mycoplasma. So they did the comparative analysis. So these are the two ways they looked at the data. One is they gave the whole set of information about organism genome. Also they gave the function. In another paper, they gave the gene map function as well as they made the comparison. That's what exactly we are doing even now. So since 1995, we have been using the same in all our genome analysis of any microorganism. So that is the basis of everything. In 1996 or 97, there was another paper that was in 96. So they also predicted about the minimal set of cellular life derived by comparison of complete bacterial genomes. So one of the group, they actually use the information available in the science paper about the Haemophilus and Mycoplasma. They came out with that, we need about 256 genes, which are considered to be the minimal set of genes required for any bacterial organism to be alive, viable, able to grow. So that was beautiful information, but they did the comparison of just only 2 organisms. So those were actually published in 1995. They used that. And then, people started sequencing several other organisms. I'm just giving you in earlier times, there were actually 74 laboratories which were involved in sequencing yeast, Saccharomyces cerevisiae, which began in 1985, 89, and 1996. So 7 years, they were able, they involved 78 laboratories to sequence Saccharomyces cerevisiae. Okay, so you can understand the amount of hard work which went into genome sequencing of yeast organisms. So, but it took so much time because the genome is almost 60 times more than the organisms like Haemophilus and Mycoplasma. So right now we have so many technologies available. In those days it was all Sanger, and then now we have pyrosequencing, ICAR and Illumina. So that is all till 2005 and 2010. And then now we have also backbio and nanopore techniques which are available for sequencing. So we have advanced our evolution of sequencing technologies. So we have several methods now available for sequencing DNA. So that makes things easier to us. So these are the list of methods which were available, developed in between. So it is 454 by pyrosequencing, ICAR and Illumina solid, and then DNA nanoball sequencing, helicose single molecule sequencing, backbio single molecule real-time sequencing, SMRT technology, nanopore DNA. So there are so many methodologies which are available. I only can give you an analogy. If you have a Maruti car and if you meet with an accident, what will happen to your car and to yourself? Just imagine. But at the same time, if you have a BMW and meet with an accident, what are the chances of you having problem? If you really want to imagine, or if you don't want to imagine, then you can ask Rishabh Pant, cricketer, about his BMW and his accident. So you can understand about the impact which you'll have. So if you have a methodology which is very cheap, probably you will commit more mistakes. If you have a robust methodology, the amount of genome sequences with the mistakes will be very, very less. So that is also very important because now there are a lot of information about the genome sequences. So we have so many organisms which are sequenced. But again, if you look at the growth of genome database, there are so many organisms, more than about 1 million organisms which are getting sequenced. But the condition is a sequence is considered to be complete if you have all chromosomes fully assembled with the gaps not exceeding 10 ambiguous bases. So if your accuracy is not then this condition, then your genome sequencing is considered to be incomplete. So we have a lot of information about scaffold, contic, in the databases, but complete genome sequencing is difficult to achieve. That is could be done only by so many trial and error. So we have now a reference sequence like NCBI. I will also come back to NCBI later, but right now I'll say we have a reference sequence RefSeq, which is having a database where they have a total number of complete genomes added or uploaded in the database in the early increments. Also the number of new updated completed in every year, which is almost 10,000. So every year we have so many sequences are going. So although we consider like sequencing is going to be really a challenge one, but there are several achievements which are available. So right now NCBI has got this many number of organisms, genomes are available. So sequencing has become so fast now. So what do we do with this kind of information? This information is very, very useful for us because our knowledge about organisms cannot be limited to certain genes. If you know the whole genome of the organism, then you can compare them and see the differences between two organisms at the strain level. So that becomes easier. So if you look at the genome from a bottom in a longer perspective or a higher level, then you may not even see those differences. But if you know at the genome, at the gene level, you may not even see the differences. But if you look at the whole genome, then it is possible to understand about the what you call the differences. There are now there are now methods about like how to compare the organisms in a better manner. And in fact, like if you look at the information which are available to us, nearly 70 to 80% of the genes in each genome are highly conserved in evolution. So we have almost 70 to 80% which are running through all the organisms in the same manner. But we have also genes which are called like homologous relationship based on orthologs, the genes which belonging to showing the similar functions are evolved in the same species. Then based on that you can understand about the functional information, also about the evolution. So we now have evolutionary genome analysis is made possible by whole genome sequencing, not just by the gene analysis. Gene analysis helps us in identifying organism, but if you really want to understand about the evolutionary relationship among the organism, this becomes very, very easy. So after sequencing, if you really wanted to compare the sequencing of genomes of organisms, we also have to have several advances or development in computational genomics, because the amount of information we get from a genome sequencing project is too much. So we need to have approaches which will identify the orthologous genes, also to utilize the genome content information for functional inferences. So we need to use that information to understand about the differences between organisms, also to predict about the functions which they can possess by looking at the gene sequences. So over the period like last 25 years, we have a rapid accumulation of genome sequences, although many of them are incompletely assembled. But actually we need to have more advances in technologies or methods and tools to replace the combination of automatic, manual, case-by-case analysis, because we don't want to have errors in our assembly, also understanding about the bacterial genomes, right? So by now you must have an idea like what is the complexities in understanding about the genome information. Even if you want to do a genome information, like you have a project to sequence the genome of your organism of interest. After sequencing it, if you do not know its function or its relationship with the existing organism for which they are available in the databases, the sequence information which you got for your organism is meaningless. So you need to know like how they are related to other organisms and how you can use the genome information for predicting the functions. So those are important. So we have now, there are many challenges. Some important paper, like you can read, which appeared in a computational and bioinformatics journal and biotechnology journal, which is about long walk to genomics, where they even said about the challenges related to faster evaluating gene families with the complex histories, also about the difficulties in horizontal gene transfers which makes things very, very complicated. So now we have, not only sequencing, that is the, what you call, technological advances which are required in sequencing the DNA, but also we should have tools for reading those sequences and understanding or inferring them from them. So we need to develop computational genomics. So I'm introducing another concept. So understand that, like, even if you do with the new technologies which are coming in the market, like backbio, nanopore, or any other method by which you can do the sequencing, but you should also know how to make use of those information. So this is one picture which will give you an interesting idea. Like, this is what you call the first early part is basically even before the introduction of the Sanger sequences, they have done it. 1965 to 75, most of the sequencing was done by, early sequencing method. And then only by 1977, it is by Sanger. From then to almost all this green color is by Sanger. No, yellow color, all this yellow color. Then it is the green colored area, 2010, 2015 is called NGS, next generation sequencing. And now we have the blue colored one, which is called third generation sequencing. First, second, and third, right? So we have first generation, second generation, third generation. Understand this aspect so that you know the power of resolution and the actual amount of correct information you are able to get from sequencing methods, okay? So, okay, but we also have now new developments in terms of single cell genomics. So what we are, we have been talking about, isolating organism, growing them, and then extracting DNA from them. Then we, do the sequencing work. But when you are growing the cultures, if the cultures are not pure, and if they have too many other organisms coming up without your knowledge, your sequence information is going to be of no use. Quality is going to be compromised. So we have now even methods available for single cell genomics, where you can select a single cell and then extract the DNA or amplify the DNA within the cell, and you can also go for the single cell sequencing. So there are methods now available, and the applications are, well, I think there is an interesting paper in Cell Bioscience where they are even given different methodologies by which you can do the single cell sequencing, right? I'm going to give you some introduction because this will be the future of genome sequencing. So if you are talking about a microorganism, if you are able to bring a single cell into sequencing, then you can be very sure that, like, you are dealing with the organism which is very pure and there are no contamination. The information is very much related to that particular type of cell. Well, so then we have now single cell amplified genomes. SACs are available. So we have methodologies where you can isolate a single organism, then do the sequencing. There is a paper which appeared in one of the journals, like Biophysical Trends, where tools for microbial single cell genomics for obtaining uncultured microbial genomes. So even for the organism which are not being cultured, you will be able to do it, because you can take any sample, any environmental niche, and then select a single organism from that sample, tissue or a soil or water or plant tissue or anything. From there you will be able to even quantify it. So this is what exactly they were proposing, like you can take any one of those environmental niche, then isolate the organism, encapsulate them, put it in a gel so that you will have a pure cell, then do the genomes amplification, isolate them, then go for the sequencing, and then you can also understand about the single cell amplified genome. Right. Then there is another way of doing it, because if you introduce Propidium monoazide, this chemical molecule can get into the cell through membrane, then it can bind to the DNA. So any organism which are dead, then this Propidium monoazide will go and bind to the DNA, and it will not be available for amplification. So even if you want to make a difference between active and live cell, you can do that here in this method. So in all other methods, WGS, in all the methods which we have discussed, we will be able to, we can't, we assume that everything is growing, they are viable. But there could be a combination of dead cell as well as viable cell. But in this case, there is a chemical to differentiate between the viable and dead cell, propedium monoazide. So with that treatment, you will be able to differentiate between the live cell and dead cell, and then you can subject the live cell into genome sequencing. So single cell assembled genome is now possible using this method. There is also a review in Nature Microbiology, I think. I'm not able to even read this paper. They have written something about single cell genome sequencing, current state of the science. They describe the methods and advantages. So if you have to read them, because by the time you become a full-fledged professional, these methods which we have been handling today will become irrelevant, because rest of the people will be doing single cell genomics outside India. So abroad they will be doing single cell, and if you are doing with your sample, then people will say, question you, what is the relevance of your sample? How can you say that, like you are dealing with the cells which are viable? So differentiate between the active cell or those dead, or even the cells which are impure. You have to go for the single cell genome sequencing. So there are methods which are given for whole genome amplification, even in that paper, in Nature paper. Something like degenerate oligonucleotide primed PCR, PCR, multiple displacement amplification, MTA, and then a combination of method called multiple annealing looping based amplification cycles and pico-efflux. So all these combinations are difficult to understand for you at this moment, but you start learning it, because even when it comes to PCR, we have so many methods, so many diversity of methods in PCR. So if you have a PCR in your laboratory, you should also know what way you can make use of that equipment. So exactly that is what Craig Venter did. When he was approached by PerkinElmer that they said they have a automated sequencing machine, he immediately jumped and got the machine from him. He in fact spent $300 million from his own saving and bought the machine, and that led into the whole exercise of publishing paper in 1995, 2 bacterial genomes, right? This guy also did another job, because he collaborated by then. He published an important paper called Sequence of Human Genome. If you look at the number of authors, it looks like it itself is a genome sequence. So many authors are there. So that paper appeared in 2001. So he didn't stop with the bacterial genome. He jumped into human genome. So this paper, he is the first author, and for which he got a lot of appreciation as well as criticism, because he was actually competing with National Institute of Health of US itself. So by the shotgun method, he jumped and then got the whole genome sequence and got the paper out. And he did another important thing. So I'm going to take you back to Charles Darwin. How many of you must be heard about this old personality in science, proposed everything about evolution. He went on a voyage, and that voyage, he traveled from UK using a ship which is called HM Beagle, and then he went on for 5 years. He visited Galapagos Island. He was somewhere in this between North America and South America. This island is there. He collected samples and he wrote a book on evolution. Same way, this guy also did another thing. He went on his own voyage in his own yacht, what you call ship, that is called Sorcerer. And when he came back, he published a paper in 2004 that was about metagenomics. So we jumped from microbial genomics to metagenomics, right? So what he did is environmental genome shotgun sequencing of the Sargasso Sea. So this is the yacht event, so Sorcerer. So some of you must have read the book of Harry Potter, Sorcerer Stone 1 and 2 and all. So if you want to remember that this and this has got some relationship, I don't know. Okay, so this paper was published in 2004 in Science, and he did the same approach, shotgun sequencing, got the samples from the sea water, and then he could identify more than 2,000 different microbial species in the environment. Again, one classic example of metagenomics. Of course, he didn't, actually in this method, he went by clone approach, where he prepared the clone and also looked at the different genes like 16S ribosomal RNA, elongation factor, and then what you call heat shock protein, elongation factor TU, and REC-A, and RPOB. So all those genes he used it. He didn't do the whole genome sequencing, but he used a battery of genes, and then he looked at the number of organisms which are in different phyla. It was an interesting paper. But of course, before that there was a paper by Norman Pace, who actually reported that in the environmental sample we have both culturable organism and unculturable organism. There was impact of culture-independent studies on the emerging phylogenetic view of bacterial diversity, JVAC. And this guy worked in the University of Boulder. Even now, if you wanted to know the developments in microbial genome or microbiome, go to this university website and learn about their developments, okay? But actually the word metagenome was introduced by Jo Handelsman, this lady. When she wrote a paper in chemistry and biology, she used the term like the collective genomes of all the microflora, which we term the metagenome of the soil. So that picked up, and now we are all using this word, which was actually introduced in 1999. She has an interesting review which is published in Microbiology and Molecular Biology review paper, that is metagenomics, application of genomics to uncultured microorganisms. So what exactly, then also we have now a lot of interest in microbiome, because we could now understand that organisms are associated with the plants, animals, environment. So if somebody wants to define any organism, you can't define it without microorganism. I used to ask some of the entomology students who come to me, they'll say, I used to ask them, will you define insect? They'll say, no, sir, it's difficult to define, because now we know every organism is having a combination of genetic pool of your own cells and microbial cells. So now we know that, like, you as a human being, you are not you alone. You are with your microorganisms. So that you should understand that. So genomics is now going to play an important role to know what is the difference between you and another person, not only in terms of your genetic information, but also by looking at the microbiome composition of you and the person which you wanted to compare or analyze, right? So we have metagenomic sequencings, and basically what we really want to know is, we wanted to know who are all there and what are they doing. So these are the two things which we wanted to identify. So we have several methods. We do marker gene analysis, binding and assembly, by we can look at the diversity, and then from there the genes we can also predict about the function and functional annotations. So we have comparative metagenomics, where we can compare about the intercommunity similarity and biomarker deduction. So I'm just going to go through fast, because I think I have another 15-20 minutes only. So we have two major methods, which are targeted and shotgun metagenomics. Targeted means we just wanted to study few genes. We know the genes. So we select the genes and look at the compositional differences. So we have a lot of advantages in terms of depth, preciseness, but there could be unequal amplification, then not enough resolution for species and subspecies identification in case of targeted one. Whereas a shotgun one is, theoretically you can compare, you can study all the organisms which are available in your sample, can assess the function, but greater resolution to genetic content, but actually rarely you can't do that. Rarely some sequences, everything can be sequenced in the sample, not so much of depth, and the money factor, which is very, very important. So we have targeted metagenome, we can take any sequence, barcode them, align them, qualitatively you can understand about the OTUs, and then we also do these diversity estimates in terms of rare function and common factors. I think some of you must have got all these ideas from those exposure in the practical classes about others, but again, you spend your own energy and time to learn about them, because you can ask others to clarify your doubts, but if you don't make your own understanding about this, it's going to be difficult in the rest of your life. So that also you understand. So you have to make your own efforts to learn about them. So when we prepare a metagenomic data, so we always compare in terms of alpha diversity versus beta diversity, community memberships versus community structure, and phylogenetic information versus taxon based, and also about the function. So when we talk about the alpha diversity, we actually study how many tags are in a sample, establishing the profile. If you have 2 samples, there are 4 OTUs and then there are 3 OTUs. The other one is beta diversity, how many tags are shared across samples, comparing profiles. So 3 of this and 3 of this are, this will say in terms of beta diversity. Then if you wanted to say the community structure, relative abundance in each of these OTU, how many such fragments you are able to see or OTUs. So these are some of the basic information like community membership, and also like you can prepare a pie chart to say about like what is the actual structure or the membership of those samples. These are available, okay. And we have now pipelines which are available online, and which is one such is called CHIME, although the spelling is QIIME, which is Quantitative Insights into Microbial Ecology. It's a modular pipeline for analysis of both targeted and shotgun data. So shotgun analysis will be still considered beta. So in the website is available. You can use this pipeline for analyzing your data of metagenome, right? So we have, it's a bioinformatics platform which is extendable, free, open source, and community developed, which will be used for analyzing the data of metagenomes. So we call CHIME 2. And if you look at the number of authors who are responsible for reproducible, interactive, scalable, extensible microbiome data science using CHIME 2, so many. This reference is 2019 paper, so you please read this paper for improving your understanding about the metagenomics analysis using CHIME 2. And we also have a shotgun metagenomics, what we really say in the sense, whatever we saw in the principle about 1995 paper about whole genome shotgun assembly method of Greg Wender, the same thing which we are using. So I'm not going to repeat them, but what is important is you should have a clean data. If you have some kind of a contamination in terms of RNA, human, and there are problems, like some of the uninformative reads like low quality, low complexity, artifacts, adapters have to be removed. So we have to remove the contaminants using some of those pipelines like CD HIT or UCLS. And you have to remove all those things before you start your analysis. Then look at them, remove the low quality bases, low complexity sequences, and adapter sequences. And there are pipelines like PrintSeq, which will integrate data and generate quality statistics, trim, filter, mask, and dust. So this so now you are jumping into another field. So whatever you know about your entomology, pathology, microbiology has to be complemented by the knowledge about bioinformatics. Otherwise, you won't be able to make a good progress in your analysis. So there are ways of removing the contaminants, host sequences, and all those things. And this is, that makes a lot of difference in the final analysis. This is one which is before the contaminant removal, which will give you a distribution in a different way. But the moment you remove all the, non-hits, removal of contaminants, the composition completely changed. So much of importance is required for removing all those unwanted sequences. Well, I'm introducing one more, Kraken, which is a pipeline which is available for analyzing the data. So that is again using built-in databases of RefSec related to bacteria, viruses, plasmid, even for the human. And fungal database is integrated into that. That can be used for analyzing your data. So genome sequencing or metagenomic sequencing or microbiome analysis. Well, for genome sequencing, we leave it. Microbiome analysis, we can use that. Kraken will depend on the queries which are based on the KMS, and then they will prepare the taxonomic tree, and also you can understand about the classification. So Corona is used for actually visualizing the Kraken. So if you look at some of the papers now which are published on microbiome or environmental genome, you will be able to see this kind of a picture coming up. So these are nothing but Corona, which is actually available in Kraken. So CHIME, Kraken. So there are several other pipelines. They are available online. So you and I have to not to choose only chime and kraken. So you can choose whatever you want. It's like, there are so many variants of car which are available in the market. So you don't limit yourself to Ambassador, Padmini, Premier Padmini or Maruti 100. You have so many variants now. You can take depending on the money which you have, or the access you have, or the comfort level you have in analyzing your data. So to summarize, we have targeted metagenomics, shotgun metagenomics. Also we can do the functional analysis using shotgun metagenomics. So targeted means we will select the panel of genes, target them, amplify them, sequence them, and understand about the differences. Whereas shotgun, use the whole DNA, prepare the fragments, and then sequence them, do the assembly, and analyze them, and look at the data. So these are the two approaches. So, well, again, back to our hero, Craig. Okay, so what did he do? He didn't stop just with the sequencing of bacteria or human genome. What he even did is genome transplantation in bacteria. He selected two organisms and he removed the genome of one organism, prepared only the shell. Just imagine a bacterial cell not having a genome, that is something like shell. And he took out the genome of another organism, transferred to the organism. So he migrated from recombinant DNA technology to genome transplantation, removing the genome of an organism and putting up a new genome. So that is what he did in 2000, I think it's 2010 or 11. And also what he did is he completely synthesized the DNA by chemical means. Nucleic acid was not extracted, not fragmented from another organism. He did like you do it with the primer. He did the whole DNA synthesized in the chemistry laboratory, took the DNA, put it in the organism. So he played with all microbes, right? And that was like complete chemical synthesis, assembly, cloning of a Mycoplasma genitalium genome. So this was these two did. So we have a lot of other challenges, although we are all, we imagine, or rather we assume that genetic information is the, DNA is going to determine about the phenotypes. I'm giving an example. There's the same organism like Serratia, so Serratia marcens. So if you grow it at room temperature, this will be red in color. But if you grow the same organism cells and then incubate at 37, this will become no pigments. It is yellow in color. So what is the difference? So the same cells from the same source, you plate it in a medium and then incubate at 28, you will be able to see the red color pigmentations. Okay, but the same one, if it is incubated at 37, there is no, but the DNA is not changed. Phenotype, color has changed. So what do you expect? So now another challenge. So although we talk in terms of genes containing all the instruction for the phenotype, genotype, deciding about the phenotype, all those things we have been talking about it, but the problem is there is something called epigenomics. Even if some of the DNA, without changing the sequence of your DNA, if there are changes in terms of methylation, then the phenotype could also change. So now we have a new challenge to understand why some organisms showing a particular phenotype in a particular environmental condition, or it will respond to a different stimuli and give you a different phenotype. So there are challenges in terms of understanding about backmeth. There is a way of understanding about the methylation in bacteria, although epigenomics is being seriously investigated in all higher organisms, plants, and then human beings. But we are also now understanding about the epigenomics in bacteria. So we have to learn. Too many things to learn. So microbial genomics, metagenomics, single cell genomics, and epigenomics. Well, I take a break, then get back into another topic. So very quickly. So what are microbial identification, deposition, and culture repositories. So if you don't use your tools properly, you won't get any information. So that is an example which is given by Antoine Leeuwenhoek. He was having a microscope which is meant for looking at the textile fibers. Then he just looked at the sample, took out something from his own teeth, then in a gum area, then saw some organism. He wrote a paper and sent it to London, and he got FRS. And we are all now saying the father of microbiology is Anton van Leeuwenhoek. So if you don't have a tool, right tool, you can't do anything. From that, everything changed. From microscopy, so we have electron microscopy. It is all microscopy, but now it is nanoscopy. What is nanoscopy? So micro to nano level. So we have now, so what is called super resolution fluorescent microscopy. We also have a Nobel Prize awarded to 3 different scientists, and that is about from microscopy to nanoscopy. So we have so much of resolution and then differentiation which is possible using these technologies. And tools are now becoming toys, and of course isolating organism is also a challenge. This is one picture where father of the nation is looking at his microorganism using microscope. Of course, in those days, nanoscopy was not available, but even in our IRA, we don't have a nanoscopy. So we are still, we have to get a nanoscope, right? And well, back into this, we have several methods which we are using for isolating organism. One is culturing the organism, right? And we have now methods available for single cell isolation, something like back to tip, manual cell picking, laser ejection sorting technology. Technologies are improving so fast that if you don't know about your tools, you do not know what to use with them. But all of us know how to use mobile. Mobile Nokia in 1990s or 80s, and what you have smartphone now. The difference? You understand the difference. iPhone and Samsung, and 10 years or 20 years back, what we had in our hand is something totally different. So if you have a right tool, you can learn a lot about that. Also, another important thing which I want to talk about is depositories. So a cell isolation, any of you, can you recognize whose handwriting and what is this paper about? Well, by sitting there, you won't be able to see that, but at least you can read the name Gregory Mendel. Who is Gregory Mendel? So this is what his lab record, this guy, okay? And this is the actual lab notebook which he had. And now this is being insured for 2.5 million euros. This particular manuscript, which has been prepared by Gregory Mendel, is being insured for 2.5 million euros. And that was actually like what is written is an original manuscript written by Gregory John Mendel, and that is on experiments on plant hybrids. And there are 12 sheets in which Mendel summarized the results of his many years of experiments on heredity, and that is how important for this. 2022, we also have a bust of Gregory Mendel installed in the division of genetics. If you have an opportunity in the evening, go to the genetics division, look at this bust Gregory Mendel's, which is kept in our own institute. So that was celebrated in 2022. So actually, when he wrote the paper for 35 years, nobody bothered to give any importance to that paper. So it was lost. That one later, 35 years later, only they realized that his experiments are useful, and then they could understand about the laws of Mendel, right? And this is another lab record book, a record of another famous scientist. Any of you can guess this? It's again by RNA sequencing of Frederick Sanger. Sanger, who got 2 Nobel Prizes. When he was 40, he got 1 for protein. When he was 62, he got another 1. That was about 2 manuscripts. And again, this is another interesting lab notebook of one more scientist, Carey Mullis. Why I'm showing all these lab notebooks? How many of you bother to keep your own lab notebook safely and write everything what you do in your lab notebook? It's a question. Good, I like it. So unless you take your record writing, lab maintenance is very seriously, sincerely, that all this, doing experiments are meaningless, right? So keep a lab notebook. Why do we have to talk about the lab notebooks? So this is the beginning of all those things. So if you give this much importance for your own work, and if you have a lab notebook, what about maintaining your cultures? it's also important, no? Whatever you use as an experimental material, lab study material, should also be maintained. Whether it is an insect or a pathogen or a microorganism, whatever you do, you should also have the. So this is Karimullis. Actually, I want to say that when he wrote this paper, his manuscript was rejected by Science, Nature, and it was published only in Methods in Enzymology in 1987 about the PCR technology. But in the end, Nature came out with the issue highlighting his work, again, Karimullis. So even if your paper is not published, but if you are honest with your work, well, you will get your own recognition. you have a chance to get somewhere appreciated. So this is about the notebooks. Back to the when Mendel wrote all his paper, it was in 1822 or 1884. Just imagine when we don't have even properly notebooks. We have now so many books are available in the market. We have tools for writing. How many of us write? We don't write. How many of us will keep our lab notebook safely? We don't. Well, there are challenges, but I will share another information. When I happened to be in German laboratory in Max Planck Institute, the director of the institute told me that after I leave, my lab notebooks will be in the vault of the institute for 10 years. After 10 years, if there are no more developments and if they consider that everything is published or there is no more need for using any information from my lab notebook, they will be destroyed. Here, we don't keep the lab notebook. The day we leave, I don't know where else it will go end up. So that is the importance for the lab notebook. So back in 1911, there was a guy called Frank Karl of Prague. He was the pioneer to collect the cultures and then he made them available for free, bacteria culture. So that was the culture beginning, the repository was set up. So that it was in Vienna, then it was also we had another oldest collection, Central Bureau of Cultures in Netherlands. So bacterial culture collection, the importance was understood in 1910s, 11 around, okay. So this is a guy who was a pioneer in doing that. So now I'm going to go through the slides quickly so that you have an understanding about all of them. So we have a World Data Center for Microorganisms. Visit the website and understand about that. So initially it was in Australia, but now all this work, all this data management is with the Chinese. So if, I've been told by my professors back 20 years earlier that when they were students, they were asked to learn a foreign language. Either they should learn German or French or even Latin, Spanish. Okay, and I warn you, if you don't do well, there will be a time where you will be asked to learn Chinese, because now everything is maintained by these guys. So if you don't take things seriously, it's not Hindi. You will be asked to learn Chinese to understand science. Well, this WTCM is having a culture collection registers info, culture collection information worldwide. So number of microorganisms which they have information. Do you really want to do all genome sequencing for all these organisms? Where is the money? Show me the money. Okay. So we have a global catalog of microorganisms. So many strains are available, so many nucleotides are available, literatures are available. So whatever you do, you have to do by looking at what is what is available elsewhere. Compare your work with others so that you know what you are doing is something novel, something new, or you are repeating the same thing which are available already in the databases. So most of the informations are now available from China, then Germany and US and all those things about the global catalog microorganism. So we have information related to whether they are plant associated or host associated from different animals, soil, and also the number of strains in E. coli, Aspergillus. So all these things are now available in the internet. So www, World Wide Web. So we have reference strain catalog where also we have something about the strains which you could use for phylogenetic analysis. So there is a International Union of Microbiological Societies which is there in IUMS, which is collecting all the connecting to all the national societies, and they also have information about all the microorganisms, right? So they have divisions called bacteriology and applied microbiology, mycology and eukaryotic microbiology, and virology sections. Okay, they have they also conduct workshops, they publish, they have culture information. Recently they came out with a paper called "Save the Microbes to Save the Planet." If you really want to know about the microbial role in the life process or on the earth, read this paper and understand about the importance of this microorganism. The national level, we have a National Collection of Industrial Microorganisms, NCI, National Chemical Laboratory, NCL, Pune. So we have one which is actually a member of World Federation of Culture Collections as an online searchable database of strains. And they have also offer you service you can buy cultures from them. You can also get your cultures identified by them, and then you have they charge you for that. They'll do the phylogenetic analysis. They also do the biochemical analysis. These are available. Then there is a national facility for culture collection of fungi, which is there in Agarkar Research Institute, again in Pune, which is having something about fungal organism. So we have one in IRA, Indian Type Culture Collection, IITCC. So you go to Division of Pathology and then have a look at that. And then there is one in Chandigarh, which is known as MTCC, that is Microbial Type Culture Collection and Gene Bank, and which is also a member of World Federation of Culture Collection. And probably this is the first IDA center. So IDA is something like international, Depository Authority, International Depository Authority, Budapest. Based on that, any culture which is maintained here you can go for patenting and all those available there, right? So read about the Budapest International Depository Authority and understand it is one of them. So we have one in ICAR system that is called National Agriculturally Important Microbial Culture Collection, NAIMCC, that is in Mau, and then where you can also get your cultures. Well, I'm going to show you something interesting. These are a culture collection which are maintained inside the deep freezer or in the freezers or in refrigerators. There is one interesting museum of microorganisms. This is probably the only museum which is available in Netherlands, where you can go and have a look at the microorganism growing on petri plates, and also organisms which are associated with human beings, human gut region, or with your oral microbiome, or with the plant system. So they have a museum called Microbia, and which is there in Amsterdam, and you can have a look at the live microorganism, something like a zoo. This concept is now coming up. So anything you wanted to learn about microorganism, visit their website. You will have many surprises. Well, I have some of us must be knowing about ATCC, American Type Culture Collection, which is also a standard one. They give you so much information about all the microorganisms of in earlier days, cultures from here were actually sent to ATCC for deposition. So then we have now in MTCC in Chandigarh. In initial days, before 60s and 70s, culture was sent actually, even from other countries, they used to send it to ATCC, which is there in Virginia, right? So we have, this website is having several interesting information. So they not only give information about the microorganism, they also give you about the media composition, bacteria and phages, and also the certified reference materials if you really wanted to compare your organism with the existing one. So we have, they also have a new genomic DNA and RNA, where they have a deposit information. So all these things will give you an idea like how seriously the work is done, how much information are deposited in one of these repositories, so that your information is safe and available to all other workers. It is not like I will see, and then I forget and throw the culture. That attitude is going to be a great loss to every one of us. So of course, all of you must be knowing about National Center for Biotechnology Information, NCBI. We all been using it, but we have come to a stage where we end up in a situation called GI-GO. What is GI-GO? So if you are sending garbage in GI, you will get garbage out. So if NCBI, if you are depositing all unverified standard information sequences, cultures, what you get out is also garbage. So that is one danger with the NCBI, because NCBI being used by several people, but they don't take care about the quality of information which are deposited. Although I say about GIGO, but there are some concerns about the information which are being deposited in this. NCBI is actually with a great idea that like we all have to learn from the alphabet to words and phrases in molecular biology, so they could understand the importance of the DNA. So they started this, DNA emerges as a syntax of life process whose most complex expression is man. From there, that was the mission of having NCBI. In Nucleic Acid Research Journal, they also submit about the database resources of the National Center of Biotechnology Information. So they give you about the PubMed and then resources, literature, and genes and all this information. So every year you can go into nucleic acid research towards December, January, they used to come out with the issue, and then they gain information about all those database bases available in the worldwide available for others to use. So we also have European Molecular Biology, EMBL, European Molecular Biology Laboratory, where European Informatics Institute is there, and there is a website which is having a lot of tools and databases, information about organisms. So we have another one called Department of Energy Joint Genome Institute. They have integrated microbial genomes and microbiomes. So that database is also useful to understand about the genome information. So as I told you, the database issue will come in probably in January of nucleic acid research, where they will mention about the online molecular biology database collection. So you can update the information which you have about all these databases. So we have something related to EMBL, which is in 2022, like in December 2022 it came, and also about we have NCBI in 2023. So now in November you will get something about this. So we have several databases. So there again, so we have genome variation, then also raw data and metadata. So we have to be really careful what we are submitting as a raw data. So that's what I said about GI garbage in. So if your raw data is not clear, then your output is also going to be questionable. So we have also information related to the biodiversity, single cell omics. So it has already taken place in the databases. So in next 10 or 15 years, this is going to be the most important than your genome. Then epigenomics, well, you have to be ready for this, because just genome sequencing is not going to be enough, because epigenomics is going to be another important one. Then we have non-coding RNA. Some of you must have been informed or learned about non-coding RNA, where we have several RNA, like tRNA, ribosomal RNA, which are not protein or which are not involved in expression, but they play an important role in RNA processing and interference. So we have another journal called Database, which is actually by International Society for Biocuration. So they have a journal called Database, where they'll publish all the primary data, and so that's going to be really important for us. So if you are having large amount of data but not analyzed, you can also send it to this database information. So back to our own Greg. So what did he do? He didn't stop with all those experiments. What he did is he published a paper called The Diploid Genome Sequence of an Individual Human. You know who is that individual human? He himself. His own DNA was sequenced, and it is available in PLAS biology. So he is the first person whose genome is sequenced, and that is available. So he did that, and he also wrote 2 books, and one is called Life Decoded, something which is very interesting for you to read. So please go through that. So as I told you earlier, there is a paper which appeared in 2016, where they design and synthesize a minimal bacterial genome. So he was not satisfied with the diverse organisms which are already available on the planet Earth. So he designed his own microorganism. So he created a synthetic microorganism with a minimal set of genes. So that is known as Mycoplasma mycodium, I think, Mycodes JCV 1.0. like some movie like 1.0, 2 version. So we have that he's having Mycoplasma microtus JCV 1.0 and 3.0. So he has got DNA synthesized chemically in the laboratory, prepared the cell, and also he is creating a synthetic organism. So that is something which is developing. So we have come a long way from sequencing the existing organism to developing our own synthetic cells. So synthetic biology is going to become a boon or a ban which you have to design, because anything we do more, sometimes it may bring disastrous results. We don't know what is going to happen. So we have concerns about genetically engineered organisms, genetically engineered plants, but now we are talking about synthetic organisms, right? So this is thank you for your attention. After attention is left, what he wanted to say is his success came from belief in his data and approaches he was taking. So if you are not sure about the data which you have, your success is also questionable. So that's what he wanted to say for his young people. So I thank all of you for your attention. Right. With that, I'll stop. If you have any question, you can ask me. Taking 10 minutes more. Any questions, please? It's very difficult for me to not to sleep, but I could see some of you are sleeping. Well, that's one problem, when you postpone or adjust your timing of presentations, post-lunch session. So if you are given a heavy meal, your gut microorganism will play a different role, will send you to slumber, to make you sleep rather than being happy. So listening about their work is difficult, I understand. Any question? Well, even if you don't have question, please ask question, okay? That's what Greg Wonder was saying. So if you learn how to learn, that's one of the key messages he has said to all of us. This guy has not been given Nobel Prize. Understand? He has done so much, but people in the establishment didn't like his aggressive nature, so he was never considered for a Nobel Prize. He's yet to get. I do not know whether they will ever consider him for a Nobel Prize. But he did so much for molecular biology, especially once he realized that he has an automated DNA sequencer in his hand, and he went full blow, and he could sequence everything. So that's what I think I told you, he bought a sequencer for $300 million using his own money. Perkin-Elmer also collaborated with him, then he went on to do a lot of work. That only sequencing was not the work he was doing. The way he used the information was something which is very, very, I'll say, something which is unimaginable for many of us. He could do so many things. He could also think about synthetically developing organisms, synthesize DNA, insert them, do the transplantation of genome. Well, that's about his work. And then although we have been teaching you about different strategy and methods, it's all your imagination which is going to take you, or rather will bring you success. Okay, so some of you should actually use the methodologies, strategies, or some new ideas. Then only you can be successful. If you repeat the same stuff, you are not going to be successful in any way. Well, if you don't have a question, I'll go and sleep, right? Thank you. Thank you very much.
Thank you, Dr. Ramakrishnan. It's really a wonderful lecture. Some of the lectures will give some out-of-box thinking. In that way, he actually set the tone of the lecture by drawing inspiration from the history, whether it is a Gregor Mendel's notebooks or Andrew van Leeuwenhoek, and last but not the least, whole and whole, he is inspired by the great maverick, I can say, molecular biologist Greg Winter, So with all these bits and pieces of information he narrated, actually indeed he has covered 2 lectures. Dr. Archana was to take the other lecture on the microbial type cultures and others. So he put into both the lectures in 2-in-1 lectures. So really Ramakrishnan's 2.0 was very interesting. Thank you, sir, for delivering us, for benefiting our audience. So on behalf of the organizers, a small token of our love in the form of green plants. Thank you. Board nahi hai.
Thank you.
I don't like the board was there.
Thank you very much. Maybe another 10 or only 5 years we will have genome sequencing of this also. Researching to do that. Maybe it is interesting to do.
Yeah.
Thank you.
Just wait for the tea, have a break, and after the break you have one lecture, that's all. The tea will be here.